Okay, so this is going to focus on some work that I did at the beginning of my PhD research and then some more recent reanalysis of the same data using a different method. And I have 40 minutes tomorrow to talk about methods, so I'm going to not really show a lot of methods, but I encourage you to hold your questions about the methods until my talk tomorrow. So I think this work really came out of this idea that a lot of the studies going on linking climate change to changes in fish populations have been very correlative. And so it's largely because we don't have the capacity to go out and actually do experiments in the real world to see how these fish are responding to climate change. But we sort of strove to go beyond correlations when trying to think about the processes that are affecting and introducing variation into fish growth. And so to do that, we really came back to key hypotheses about what mechanisms might be driving changes in fish growth. And I think Tim and William have really set this up perfectly because they gave a really good foundation of sort of the bioenergetic framework about growth and sort of theoretical predictions. And so this is more about how do we transition from what happens in the lab to what happens in the real world. So what drives fish size? I'm going to talk about three kind of key components. The first is food supply. So obviously if you have more to eat, you're going to be able to be bigger. If you have less to eat, you're going to be smaller. And so there's a lot of evidence and theoretical predictions about density-dependent growth and that competition would drive changes in fish size. Largely with conspecifics. There's also this idea of bottom-up productivity having a role in fish size. So we know that the oceans are patchy in time and space, and at certain times there's more upwelling, there's more nutrients, and there's more for fish to eat because of just the inherent productivity of the system. The second component, of course, is physiology. This is a graph, picture showing, I think, that El Nino Southern Oscillation and how that introduces changes in sea surface temperature and anomalies from mean temperature. And we know that growth is a metabolic process, and so like all metabolic processes, temperature is going to have a really big impact on that growth process. So if it's colder, everyone's metabolism slows down. If it's warmer, all of the ectotherms' metabolism is going to speed up. And the thing we haven't talked about as much are top-down effects. So I think it's really important for us to remember that what we see when we look at a sample of fish in the ocean is, as Tim said, not just the product of growth, but also the product of mortality. And mortality is not uniform across size. We've got two top-down pressures that are acting in different size selective ways. The first, and probably by maybe the most powerful when we're talking about fish species, is fishing. So fishing is size selective. It tends to remove the largest and fastest growing fish from the population. And so that's going to affect what growth and size look like in your sample. The second is predation. And so fish are generally gate limited. They can't eat something that's bigger than their mouth. And so if you're smaller, you're more vulnerable to predation. So there's an evolutionary pressure to grow bigger because smaller individuals are more vulnerable. And so we can't actually see any of those processes and mechanisms happening in the ocean most of the time. And so what I'm going to do is talk about using empirical data to try to infer if patterns we see across stocks and space are consistent with hypotheses that refer to different mechanisms. And so the first part of this is looking across a large number of stocks and actually three large marine ecosystems. So I'm going to talk, again, not really about the methods, but we looked at 37 different fish populations of demersal fish across three large marine ecosystems on the west coast of the US. And so this is Alaska, this is Canada, this is the western coast of the US. And the offshore regions corresponding to the exclusive economic zone there are the eastern Bering Sea and Aleutian Islands, the Gulf of Alaska, and the California Current. And we fit state space models to empirical data of mean length at age. And state-space models are really neat because they allow you to separate process error from observation error. And so we have this autoregressive process that represents the true population mean length. And so the mean length of the population at a given age in time t is dependent on the mean length of that cohort in the previous year. But what we have to keep in mind is we don't actually see this true process. What we see are sample means. And so this is going to be affected by changes in sampling over time. And as you can see, the sample means are not inherently temporally correlated, other than the fact that they are deriving from this temporally correlated process. And so this structure allows you to better get at the difference between what's happening in your observations and what's happening in the true underlying process. And across our 37 populations that we looked at, we saw 3 key types of patterns. And so I'm going to show you a couple graphs. In all of these graphs, the x-axis is time, the y-axis is the normalized deviation from mean growth, or mean length at age, and then the lines represent our median of the Bayesian posterior, and the blue or different color shaded areas are the credible intervals, the uncertainty around those observed patterns. So in 4 of our 37 stocks, we saw a clear trend. And these trends were not necessarily always downward over time. And so what I'm calling a trend here is that it's pretty clear that the deviations are sort of grouped either above or below the line in the beginning, and then they switch to being the opposite in the end. So for some species, these were decreases, like this is Gulf of Alaska Pacific halibut, and for other species, these were increases over time. Far more commonly, we saw a much more mixed picture, and I'm calling these interannual patterns. And this is sort of where, as you can see in this case for Alaskan walleye pollock, you don't see a clear trend over time. You see kind of a lot of bouncing around, but you can't necessarily say, oh, in the beginning they were smaller and now they're bigger, or vice versa. And that was by far the most common pattern we saw. 4 species actually had an interesting result, which is that model selection supported the idea that their growth did vary, but when you actually looked at the trends over time, it didn't really look like there was that much that was significantly different from 0. And a case of this is California current chili pepper rockfish. And so for these, we just excluded these from our conclusion that growth varied, even though model selection said that this was a model that fit better. So in summary, growth variation, I would say, was pretty common. A little bit less than half, so 13 stocks out of 37 showed either those trend-like differences from average growth or those interannual differences from average growth. However, when we looked across the California current or the Gulf of Alaska or the Eastern Bering Sea, there was no clear pattern. Every species seemed to be doing its own thing. There wasn't a lot of, oh, everything's getting smaller in Alaska at this time or getting bigger in Alaska at different times. So we didn't really feel like we could try to correlate this to anything because, as I said, we wanted to not just be correlative. So if we didn't see that signature, as Tara was calling it, across different species, we didn't correlate it to any driver. But one thing we thought about is, hey, these large marine ecosystems are really big. I mean, we're aggregating a ton of data across large spatial gradients. And so as we know, it's commonly shown that growth varies latitudinally. And there's also a lot of little, eddies and currents that sort of create localized conditions that might favor better growth or poorer growth in different areas. So we thought maybe the issue is that we're looking at too broad of a scale. And because we're looking across a huge, large marine ecosystem, we're also not considering all the different changes in sampling that occur spatially. And so then, this is very preliminary work, I should say, so I'm definitely interested in you guys' feedback on this. Recently, we've been using Jim Thorson's BAST package, which he talked about yesterday, to model mean size at age across the same stocks, or a subset of those stocks, but also taking into account spatio and spatiotemporal covariation. And I'm not, again, not going into the methods, but they're summed up in his paper introducing this package. So we looked at a couple different alternative model structures. We looked at including either only spatial covariation or spatial and spatiotemporal covariation. And we looked at two covariates to start, temperature and depth, and we have readings of what the temperature and depth were at each toe of the data. I also want to look at density, but I've only done that yet for one species, so I'm not including it here. And so we started zooming in on the Eastern Bering Sea and Aleutian Islands large marine ecosystem I introduced earlier. And the reason for this is that this system really had the best coverage of fishery independent survey data going back for the longest time series. And I've applied this so far to 4 of the stocks out of my original 37 in this analysis. Pacific cod and walleye pollock, which are kind of the big 2 commercial species in this region. And then also northern rock sole and yellowfin sole, which are also commercially important, but maybe not as focal. And I had to choose an age of each species to model growth in, and so I just chose the age that had the largest sample size. So our model selection results are shown here. For 2 species, Pacific cod and walleye pollock, spatiotemporal covariation was supported by model selection. And for 3 species, depth was selected as a covariate. So northern rock sole, yellowfin sole, and walleye pollock. And you'll notice here that temperature wasn't significant for any of these species. And of course, I think we have to keep in mind, as Joey pointed out, the covariate for temperature we have right now is just the toe temperature, not the integrative temperature that fish has experienced. So that could be why we didn't see a correlation. I also think it was important, maybe go back. I think it's important to think about also, when I was thinking about why temperature wasn't selected, I realized the spatiotemporal covariance could be part of why temperature isn't selected, because it could be that this covariance is actually picking up all of the temperature effects, because temperature is a spatiotemporal process. And so then I wanted to look at the spatiotemporal covariance before ruling out temperature. So here is a plot of the spatiotemporal covariance by year for Pacific cod. And in each of these maps of the Eastern Bering Sea, the warmer colors represent larger fish, and the cooler colors represent smaller fish. And this really highlights a dominant feature of the Eastern Bering Sea, which I think is important to keep in mind, which is the shelf. And so across pretty much all species, the Bering Sea shelf kind of goes like this, and it gets deeper. So across all species, cross-shelf variation is almost sometimes stronger than north to south variation when you're looking at growth. And there does seem to be kind of a latitudinal gradient, maybe, if you look at the outer edge of the shelf, but that cross-shelf difference or that deepening is almost stronger than the latitudinal gradient in the system. So looking at this, there definitely do seem to be patterns, most notably for Pacific cod. It looks like they're bigger in recent years than they had been in the past. And so this could be pointing to an effect of temperature, as the past couple of years have been very warm. And so if they were responding positively to increasing temperature, they might be bigger. However, I think the other thing that's important to keep in mind is that Pacific cod are sort of a big deal right now because they're not doing so well. So I think I would want to look at density before concluding that they aren't bigger because of some density-dependent release from predation as the population size decreases. When we look at walleye pollock, if anything, there are kind of cross-shelf differences or even stronger. And very interestingly, pollock seem to be the only species for which they're kind of bigger as you go shallower, and then they get smaller as you head out on the shelf edge. And it's important to realize I'm talking here about the length at a specific age. So the older pollock, which are bigger, are definitely out on the shelf edge. But within a single age, pollock of that age found across the shelf are bigger at that age as you get closer to shore. And this is also, I think here we can't necessarily rule out a temperature effect, because we know in the Bering Sea, the last couple years, 2015 to 2017, have been pretty warm. And we also know that 2000 to 2005 was a pretty warm period. And you can see that corresponds roughly to periods of poor pollock growth. However, I don't know if it's, again, these are preliminary results, so I'm not really willing to conclude that yet. And then looking for shared patterns. This seemed a little bit more promising than our previous analysis at the scale of a large marine ecosystem, in that there are a couple, just squinting at this, you can see that over time, mean growth does seem to have some shared patterns across species. Notably, like northern rock sole and Pacific cod seem to be bigger in recent years. And then looking at that quantitatively, if you just do a Pearson's correlation on those indices, you can see that there are small positive correlations between a number of these species. And I put stars, so this is just a correlation matrix. And so the order of the species is the same across the top and down the rows. I put stars next to pollock, sole, and northern rock sole because I think it's important to remember those are the three that include depth as a covariate. So you would expect perhaps some correlation just if they're including the same covariate. So I think these two values might be, we should think more about whether that's driven by the fact that by the depth or because the patterns are actually correlated. But even across species that do not share the depth covariate, like Pacific cod, there seem to be some positive correlations. And notably, a very strong, not very strong, but stronger than the other weak correlations, there's a negative correlation between Pacific cod and northern rock sole. And so this is, I think looking at this, accounting for spatiotemporal covariation gives me a little more confidence in looking at correlations between species here. So in summary, I would think it's clear that there's a lot of growth variation in some of the Northeast Pacific groundfish I looked at. However, when you look at the scale of a large marine ecosystem, it's not really a slam dunk that there is consistent patterns. However, when you consider spatial and spatiotemporal covariance, there is some slight moderate correlation. And I'm not sure, again, I'm still preliminary analysis, but I'm not willing to say yet that these are environmentally driven, but at least for pollock, it seems like there might be some indication that temperature could be playing a role. And I'll take any questions.
Charged.
Probably an unfair question. How then do you disentangle the effects of environmental temperature and even the spatial temporal effects, density dependence? You know, abundance can change across the area, just not in total.
Yeah, and so I've done it so the way that I have planned to do it, and Jim is also doing some of this, fitting the same VAS package to density yields a prediction of spatial density of fish at each point, and then using that as a predictor is how I'm planning on doing that. I've only done it for pollock and it wasn't significant, but I'm planning to do it for the other species in the analysis as well. And then Jim, because it supports multivariate responses, he's actually fitting the length and the density at the same time and estimating the covariance matrix between those two for a number of other species. So that's kind of how we're planning on looking at it. So we are planning on including the spatial density as well. The only thing is we have fewer ages. When we estimate, we can estimate age structure density, but then we have fewer data. And so I've been using the whole population density, whereas I think it'd be a little bit better if you could look at like density of H6 pollock, since that's what I'm modeling.
You can tell Jim that we think our vast name is incredible. For his software, it's sounding very vast. Asta, was that you asking a question?
Yeah. Yes, thanks. I'm not sure how well this is working, clearly, so far away.
We hear you.
Yeah, we hear you.
It was a really interesting talk. I was just, maybe I missed because the internet was breaking a bit. What age at length? So are you looking at the growth or size of which size classes? Are these young ones or old ones or juveniles, adults, or across all of them?
Yeah, so I kind of glazed over that quickly. But for the first analysis at the large marine ecosystem scale, I looked at all of the ages that were well represented in the data. And so that ended up being kind of age at recruitment to the fishery to some moderately old age. And then for the second analysis, where I incorporated the spatiotemporal covariation, I was just looking at a single age. And I don't know if you can still see the screen, but I wrote it on this slide. And so, yeah, so this is just for the spatiotemporal covariation part, the ages I looked at are here. And that was driven by sample size and also a little bit of the idea that a Pacific cod at age 4 and walleye pollock at age 6 are kind of about the same size. So if we wanted to do cross-species comparison, that it would be good to look at similar sized organism.
Okay, one more question.
Because
Oh, sorry, Asta. I think Asta had more to add.
no, it's just if you. I was just wondering if we look across all ages, yet responses are, let's say, different, or even the opposite in small individuals versus large ones. Is there somehow we get the confounded effect?
Yeah, I think that is definitely an important consideration, and I did see some differences between the first analysis and the second analysis in the same species, and I don't know how much of that is because I'm looking at growth patterns in all ages versus one species. I also looked at, before I decided to only use one age, I looked at mean length of age across species and made sure that there appeared to be similar patterns across different ages before picking the one that I did.
Okay, good. So on to one final quick question.
Yeah, thank you. It's nice to hear that things are complicated. You mentioned an element that fishing mortality and the magnitude of the fishing mortality affects the actual length frequency we observe later on within the same generation time. Have you got any sense of how big that factor could be? I know it wasn't a major part of this talk, but I think it will become important when we start looking at some of the North Sea data, where certainly the fishing mortality has drastically reduced in the recent decades across stocks.
Yeah, definitely that's really important to consider, and I plan to include that as a covariate later on for the second part of the analysis. I just have sort of qualitative sense right now when I looked at the results from the first part and was trying to look at shared patterns within an ecosystem. The only thing that maybe came out is during the 80s and 90s off the coast of California and Washington, we had really high fishing pressure, and there did seem to be a little bit of a lag across multiple species at that time, and I think it was because there was such high fishing pressure in that time period. And so I think definitely in the California current, I think it definitely played a role in the 80s and 90s, and then you see sizes coming back up now that we've reduced fishing pressure.